So howdy again, my name is Daniel Kwong. This is my research over the previous summer over my RU during the Department of Plant Pathology and Microbiology tournament with Dr. Rochelle here at Texas A&M. My research is titled Nascent Diagnostic and Assessment Protocols and Explorations of Hop Latent Viroid Detection in Hemp. So first off, what is hop latent viroid and why hop latent viroid? So first off, viroids are pathogenic circular RNA and they're a unique infectious agent that are comprised solely of genetic material. They're very highly contagious and they are transmitted easily by mechanical means through unsanitation works as well as from water to insects and and also vectors. HLVd significantly impacts yield and quality because of its symptoms, which are asymptomatic until its latency period ends towards its maturities. Those symptoms including leaf chlorosis, stunted growth, as well as malformation. And furthermore, little differences between viruses and the viroids. Viruses, they have an outside protective layer. They infect all organisms and types, so animals and humans and plants, as well as they make virus-specific proteins. Comparing that to viroids, they don't have a protective layer, they infect only plants, and they don't reproduce proteins. Further along, viroid structures, they have multiple different structures, which include rod, branch, and hybrid. And something unique about hop latent viroid is that they can have a secondary structure throughout its maturity. So 1 week it could be rod, and a couple weeks it could be branch or hybrid, which makes it even more hardly to contain. So further along, industry estimates suggest that HLVd alone contributes to a market value loss exceeding $4 billion annually. This data was found in 2021. And those losses could be tied to the hemp industry, which involves cosmetics, textiles, as well as food, as well as further research shows that hop latent viroid can affect more plants than just hemp. Those include any hop-related plants, such as beer plants, as well as even very distant relatives of those plants, such as potatoes, tomatoes, or even roses. So despite these well-documented yield reductions linked to dead disease or the disease caused by the stunted growths, there remains limited factual scientific guidance available for growers to manage HLVd effectively. So our research goals, so currently viroids and other HLVd and other viroids can only be detected through molecular tests that identify the presence of DNA and RNA. With those tests, PCR's test stands for the most sensitive and accurate method, providing both diagnosis and viral quantification. Exploring more options near infrared or NRI technology shows promise as an early stage detection for HLVd. And my main goal throughout my RU was to optimize PCR diagnosis to identify symptom variations across different hemp varieties and explore alternative, cost-effective, and more efficient ways to detect HLVd at earlier stages before symptoms manifest. So my experimental setup. We focus a lot during the summer through plant maintenance, as foundational plant maintenance is key to having very constant and very replicable data. So with my experiment, we had 8 plants in total, 4 controls and 4 inoculated, and the varieties we used were titled T1 Trump and Berry Blossom. They were all around 2 to 3 weeks of age, all comparable in size. And this is just a key consistency within the project. With our project itself, how we inoculated plant, we picked the 2 leaves on the 4th node. We didn't pick leaves on anything below because we didn't want to have any soil contaminants, and we didn't pick anything too high just in case the leaf was too young. And then we sprinkled with a metal dust. We used carborundum dust to sprinkle onto create microabrasions, so when we inoculated with the sap, it was just more consistent. Further along with our experiments, this is the process of us collecting the leaf tissue all the way to RNA extraction, cDNA synthesis, PCR, gel electrophoresis, and we're going to compare that to NRI. This process itself can take 2 to 3 days if you just work consistently on it, and it's very costly as well as very labor intensive. Comparing that to just using NRI, if our data is correct and hopeful, it could be as easy as scanning the plant with a barcode scanner, and that can tell if a plant is if a plant is infected or just a negative plant. So using our results from our NRI, the greens is our absorbance and our reds and purple is reflectance. So if you see towards the 1700 range, there is an inverse relationship between the healthy and the inoculated. We want to see if that data is a probable source that shows if there is an actual differences between a healthy plant and an inoculated plant. This kind of goes along with our research and the theme of this year's symposium, the data from field to data, as if we can innovate this, it can be easy as a farmer going out to their field and scanning it with data we have, and it will be way more cheaper as well as less labor intensive than just needing to send these samples to a research lab and for us to like do that research with PCR and things, as the average farmer aren't able to do in their farms. So going to our second results, this is just showing how our plant maintenance was improved upon, because the previous summer before mine, we weren't able to get any budding with the plants itself. And on that right side, it shows a control plant with the flower forming a budding, which is very important and very nice for our research. This is also comparing the two plants, the control and inoculated, showing just the differences in between the symptoms. So as you can see, the leaf chlorosis and the stunted growth. So our preliminary conclusions, so our improved cultivation practices led to more of a consistent plant quality, as well as increased blood formation, which is very important, as we weren't able to do this last summer, shown by our increase in performance of plant maintenance. And then preliminary data shows that phenotypic variations among HLVd infected hemp varieties can also indicate symptoms may vary by cultivar. We show that there's different phenotypes with the varieties that are inoculated, which there's no research in, which is interesting, as well as near-infrared technology shows potential as a cost-effective and efficient tool for early HLVd detection, which can improve disease management and mitigation strategies across the hemp industry. So looking forward, we want to scan non-infected and infected material with an NRI machine that can see more wavelengths, just to see if we can see any more variations between both the healthy and inoculated or diseased plant, as a lot of research hasn't been done so far, as well as HLV inoculation. We want to inoculate them with different varieties just to see if there's any more phenotypic variation between them, as well as there is potential future research in plant tissue culture, as our contributors, Genetics, they graciously donated some of their material so we can scan them for HLVd, as well as we want to see if we can use any beneficial microorganisms, such as any fungus or bacteria, to see if it can just benefit the plant at all. And then we want to study research more potential ways to identify HLVd in early stages in the plant's growth, just so we can, those farmers, let's say when they grow the plant to maturity, they don't have to kill off the plants or eliminate them because the plant was later on found to be exposed to that viroid. And then these are my acknowledgement references. I would like to thank Reagan and the Verschow Lab as my main contributors, as well as Juan Beauchamp, which is the head of this RU project, as well as Genetics, our contributor, and Rare Earth Genomics, because they are one of our benefactors. Thank you. Any questions?
Questions? Yes. If viroids don't have proteins, how do they replicate?
So how viroids replicate without any proteins, so they hijack their host, so the host being, for this example, hemp, they hijack one of its cells, their RNA polymerase, and by that they're able to use that as their host and to replicate without using their own proteins in that host to make their own new tissue, new copies of itself.
One more question.
Great talk. So have you guys pursued looking at whether the NIR spectra that you're getting are more indicative of the overall health of the plant versus the actual inoculation? Mm-hmm. Like, have you compared plants with similar symptoms but without the inoculation? Thank you.
So our research, so that's something we do want to actually see if this our data is correct and that these are actual differences in a healthy and inoculated plant. So if this proves right, or if we're able to do these on different plants, we would want to do it with things that are able to be infected with HLVd, such as those roses that are rare, which is because no one knew that in the first place. So we would want to do those tests with different plants with NRI.
Any other question?
So have you seen any other confounding factors such as drought, cold stress that might look like, this disease, but which isn't the disease that could, mess up your results?
So that's a great question. So in our lab, it is controlled environment. So we keep all the temperatures and other factors that could affect the plant's look or growth within our lab. So because of the controlled environment that the plants are grown in, we would like to say that those factors of like temperature or drought or water are eliminated. So we are just mainly focusing on the symptoms of the viroid. Yes, so in the field, because of our the space, we weren't able to research that in the field, and we want to create more, I guess, foundational data in this. So when it is implemented in the field, we would like to compare those two.
Any question?
Sorry. I believe that you mentioned that in some cases plants were asymptomatic, meaning that the pathogen is there, but you don't see obvious phenotypic changes. If that is the case, would the NIR be able to tell you difference between healthy and asymptomatic plants? Thank you.
So that's a great question as well. So because that the plant being infected with HLVd can be asymptomatic all the way to its maturity stage, the reason why we are researching with NRI to see that their plant has HLVd, although it doesn't look like it has it, that's the reason why we are using NRI to see if those wavelengths are any different, or it gives us a definite point. We're saying, yes, this plant is infected with this viroid, although we're unable to see it with our eye.
One more short question.
My question is about that state where the plant is latent, or the viroid is latent. Is the viroid still infectious at that stage, or is it only spread by cuttings from a latent plant?
So those viroids are infectious based on, let's say, an insect that uses proboscis and can fly. That sap itself is what can infect other plants. So if an asymptomatic plant is near other plants that don't have the viroid and they're healthy, that insect or that vector or that sanitary means of cutting the plant and using it on a different plant itself can spread the viroid. And because it's so highly infectious and contagious, that's why it's kind of an issue, because you can't see it until way later.
Let's thank Daniel for wonderful presentation.